Hey guys, welcome to your first set of notes for chapter 17. We're going to be talking processes of evolution. This is a super cool chapter because a lot of this stuff is measurable, observable kind of concepts that are really important. And my guess is that at least a couple of these big ideas are going to find their way onto your test here at the end of the year. So I'm looking forward to this chapter. I hope you are too. Let's jump right into evolution and where we see it and how we know it's going on. So your book starts with a great example, superbug farms, and you guys might have heard the term superbug. And basically, a superbug is this catchy little term for a bacterial strain that is resistant to multiple types of antibiotics. And the fact that those exist is a great example of populations changing over time due to a stress in their environment. So they are the snapshot of evolution. And so it's not surprising that they start with these. They're a great example. And so antibiotics, you've probably been on them at one point or another in your life when you were sick. We first use these to fight dangerous bacterial diseases. Think words like uh, penicillin, amoxicillin, anything that ends in cillin, Z-Packs that you might get from your doctor when you go in for sore throats and that kind of stuff. That's an antibiotic. They kill bacteria, which is cool because we don't like a lot of bacteria sometimes, and so that works out. And the thing we learned about antibiotics that was super weird was a long time ago, we figured out that low doses of antibiotics actually helped cattle and pigs grow a little bit faster, and we weren't really sure why, and this discovery was actually kind of by accident. And so it goes really well together if you put yourself in the shoes of a cattle producer or pork producer. You want to keep a lot of animals because having a lot of animals gives you a lot of meat or milk to sell. And if your animals get sick and die, you can't sell them. And when you keep a lot of animals in a small space, disease spreads quickly. Think feedlots, think high schools and dorm rooms and nursing homes. And when someone gets sick and all these people and all these animals are in a little tiny space, disease spreads really fast. You could maybe even look at our high school as a cool example of this. So you need to kind of give them antibiotics anyway because they're all crowded in a little pen, standing on each other's piles of poo and eating out of a trough. It's not a very sanitary situation. So you give them the antibiotics, you keep them healthy, and they happen to grow a little bit faster too. It's really a win-win. So our agriculture industry in America is our heaviest user of antibiotics by far. They beat out people, doctors, hospitals, clinics, everything. They are our user of antibiotics. And so what happens with the bacteria that are exposed to these antibiotics over time is the bacteria that are not resistant to the antibiotics die, and those lucky few who are resistant survive and repopulate the gut of that animal. And so we end up getting bacteria that are resistant to the antibiotic. So what do? You make a new antibiotic. And then a few years later or months later, the bacterial strain has become resistant to that as well. And so that's how we make superbugs. And so our practice of treating these livestock with antibiotics is pretty much going to guarantee the production of antibiotic resistant bacteria. It's just that's what's going to happen. Um, Bacteria become resistant to things that are killing the bacteria in order to survive and make more bacteria. So in 2013, they did a kind of a cool investigation where it's like we're at a point in society where we know that we're giving a lot of antibiotics to animals, and maybe that should or shouldn't be done. I don't really know. So the question is a simple one. If we have a bunch of chickens full of antibiotics, by the time you kill them, butcher them, get them to the grocery store and package them, is any of that actual antibiotic resistant bacteria still in the meat? Or is the consumer essentially safe from this kind of stuff? And what they found was that about 50% of the chicken meat was infected with multiple antibiotic resistant bacteria or superbugs. And that's bad. That's really bad because even if you say, well, Mr. Stecker, that might be the case, but I deep fat fry my chicken and guarantee that that boiling hot oil is going to kill that bacteria. You know, we don't eat medium rare chicken. Well, thank goodness, okay? That's true. But in the process of preparing and cooking that chicken, you get chicken juice, from the meat on your hands, on your countertop, on your floor, and even just in the air, So bacteria can spread very easily, and so that allows people to come into contact with multiple antibiotic-resistant bacteria, and then once it infects the person, we have a bacteria that's resistant to the drugs we have in a person, and that's where we find ourselves today, of course. So, of course, we are developing new antibiotic drugs all the time. We're trying to make a drug that will kill, you know, the next superbug and keep people healthy. The problem is that the bacteria divide so fast. I mean, like E. coli is like something ridiculous, like every 17 minutes. I mean, so you can get 1,000 generations of bacteria in like no time, and they will evolve faster than we will make drugs. And so we find ourselves in an arms race between the basically the medical research field trying to make drugs to combat bacteria. And as soon as we make a new drug, if somebody misuses it or uses it when it's not needed, we can essentially undo millions of dollars in research in like a week. It's the way it works. So, superbugs are a threat to humans. We have diseases that are resistant to multiple types of antibiotics. Some are respiratory. We have here in Rapid, I know we have a lot of C. diff, which is multiple antibiotic resistant bacteria as well. And so these superbugs they're a threat. They're a threat to us, and I wouldn't say that we're not able to treat them, but we're kind of in the department where we might be losing this race in the near future. So we're keeping up right now, but we're barely keeping up is kind of the deal. So your book moves on to talk about evolution, and with this header, which is great, please don't forget this. Individuals don't evolve, populations do. That's a true deal right there. Evolution, in the strictest sense, means change over time. Individuals don't evolve, populations do. Um, you don't, go to bed one night with a house cat and wake up and your house cat has grown wings. That's not evolution. A population of cats changing size or color over, years and years and years, that's evolution. So it's not with an individual, it's with a population. So at this point in the game, if you don't know, population is a group of the same species in the same area. Species is a tricky thing in biology, and we'll get into that this chapter. Basically, if they can breed and produce viable offspring, we're going to call them a species. Preferably they should live in the same area, or else breeding can be very difficult. So that's a species. The alleles that we have in a population are shared genes. For example, you and I, the genes that code for our blood types, that sort of stuff, those alleles, those are shared genes in the human population, and that is the basis of our variation. Alleles, you'll recall, are forms of a gene, and so we have many forms of a gene, and that gives us variation. Some of you guys might be A blood, or B blood, or AB blood, or O blood. There's a lot of variation because there's alleles. And so if you have a trait with two distinct forms, that's called dimorphic. Di is two, morph is form, and so dimorphic is of course two-formed. So pea plants is one we've talked about already with some simple Mendelian genetics, having purple or white flowers. That's dimorphic. There's not a light purple or a pink. There's just purple or white. That's dimorphic. So two distinct forms is still variation. It's just not a smooth gradient of variation like you might see with some bell-curved things like we talked about eye color and things of that nature earlier in the year. So if it's not dimorphic, it is probably polymorphic. Polymorphic, poly is many, morph is form. So a many-form trait has more than two distinct forms. Blood types is the one we just talked about. You can have A positive, A negative, AB positive, AB negative, O positive, O negative, B positive, B negative. Many, many, many different forms, all coding for the same trait. The trait is what do the proteins on the surface of your red blood cell look like? And the answer is, well, it varies. It could be A antigens, B antigens, or no antigens, and so those are all different traits that we could have for that. You guys know what a mutation is already. A mutation is a change in the DNA sequence, and this is the original source of new alleles. If you subscribe to the evolutionary worldview, which you're not required to by any means, but for the sake of the test, the AP test, of course, it's written from a worldview that is accepting of evolution. It's one of the four major themes for the course. So all of the information that has changed from earliest cell to land plants, reptiles, mammals, and you and I have all been from mutations. And so those are the original source of all new alleles, at least from that view. Other events will shuffle these alleles into a variety of different combinations, and you can get a lot of different things made just by taking an allele that was somewhere and moving it to another place, transposable elements, for example, things like that. And so the combinations of alleles that you have for humans are much more than the people alive today. Your book has this number, which I can't really comprehend, but possible combinations for human alleles are 10 raised to the power of 116,446,000. So I don't know what 10 with that many zeros is after it, but it's a lot. It's really, really, really a lot. Whereas opposed to there's not even, 8 billion people alive today. So there's a lot of variety, a lot of combinations. So let's take a look at this evolutionary view of mutations. Mutation, there are a variety of types. You guys are already aware that you can have different codons that code for the same amino acid thanks to a topic called redundancy. And most of the neutral mutations you're going to see are probably going to be because of that redundancy built into our genetics and to the way that proteins are built. So a neutral mutation is a mutation. We change the DNA sequence a little bit, but we don't really see any effect on how long this organism survives, whether or not it's able to reproduce. And the majority of mutations that we're going to see in people are this type of mutation. You're going to get changes in your DNA that just happens with time, but most of them aren't going to cause much effect that we're actually going to see show up in your phenotype. Pardon me. Now, if we get a mutation early enough, or even a mutation that gives you some sort of think skin cancer or something along those lines, or some other radiation-based disease, that can be a lethal mutation. Lethal, of course, means you die from it. No duh. And then there is the rare but hopeful light at the end of the tunnel for evolution called the beneficial mutation. And a beneficial mutation is a mutation that bestows a slight advantage towards survival or reproduction. I think you'll find that as we look at the examples of beneficial mutations in your book, that a lot of them are only beneficial for a short amount of time, and that many of them tend to actually go back to the form they were at earlier. So you can kind of check that out for your own and see what you think of that, but it's kind of interesting to see that. So here you have a mutation can give us new alleles. When we cross over, we can recombine those alleles. When we independently assort, we will mix maternal and paternal chromosomes. So mom and dad's chromosomes will go to different sides. And then fertilization will combine alleles from two parents, assuming, of course, this is sexual reproduction. And then we can change the chromosome number or structure, which can be bad, but maybe if you're like a plant or something, you can kind of take that and survive it. And so that's a dramatic change in your structure or function. And so there's many genetic events that are going to give us just a ton of variety in what we see that's alive. So when we look at genetics, it is just a great tool for biologists, and it's amazing for if you're good at math. It's even better. The gene pool is all of the genes in a population taken together. Think the Human Genome Project, where they mapped it. This is all the genes that we might have, in the human population. That would be huge. You'd have to take every chromosome and figure out every single gene that's on every single chromosome, and what those genes code for. And that would be, a sample of the gene pool of the human population. And so what you do when you look at this in a population of humans, and you might want to take them from just one location to make your work a little more manageable, is you're going to look at allele frequency. which is how often does a certain form of a gene show up in my population, or the abundance of a particular allele that we're interested in. Generally, we're interested in alleles that cause disease or alleles that might confer some sort of advantage. But generally, it's disease because it gets a lot of funding and because we care about keeping people healthy, which is fair. They get the most study probably done on those. So that's the abundance among the individuals of a population. And so when we look at allele frequency, it kind of gives us our definition of microevolution. Microevolution is like we're not growing wings or lungs or gills or anything that would really quite honestly blow my mind. We're looking at changing allele frequencies in a population over time. This is actually the definition of evolution that I got when I was in college, and that's what it is: change of allele frequency of a population over time, which is not that terribly challenging to do. We can see it. We can measure it, we can see the effects of it, and we're doing it with bacteria in our guts and also in the chicken that's in my oven right now. I mean, we're doing this all the time. So microevolution is always occurring in natural populations. They're under some sort of stress, or maybe they're not under stress. Maybe they're just competing for mates, and who wins and gets the mate is changing the allele frequency just from that. These little fluctuations are happening all the time in nature. It's the way they adapt. It's the way we survive. It works. So the cool thing for the math nerds of the world, I'm not in this group. I wish I was. I envy that they're good at math. Is a way to measure evolution, and it is very cool. It's called the Hardy-Weinberg law or the Hardy-Weinberg equation, and basically it's a couple of math nerds who applied the simple rules of probability to population genetics, which is cool because like neither of them were biologists. They were like physicists and some math guy. I don't know. It's crazy. Anyways, so basically Hardy and Weinberg, these two scientists, came up with this really cool idea. They're like, what if we had a population that never changed? And here's the 5 rules they laid down. They said we had a population, hypothetical, in genetic equilibrium, which means there's no change in the gene pool. Here's the 5 rules that would have to be satisfied. You might even want to write these down. Number 1, mutations can never occur. Number 2, the population is infinitely large. Number 3, the population is isolated from all other populations, which means nobody comes in and nobody goes out. Number 4, mating is completely random. And number 5, all the individuals are going to live and they're going to produce the same number of offspring. If we can meet those 5 rules, we should have a completely stable population. Now, of course, you can probably listen to those 5 rules and go, yeah, but that's not going to work ever. In reality, that's never going to happen. And you would be exactly right. Those 5 rules are never going to happen because mating is not random and new members come in and out of populations. And the number of kids is never going to be the same. I mean, you might have 2 dogs who look the same and act the same, and one dog might have a litter of 4 puppies, and one dog might have a litter of 10 puppies, and then just like that, your equation's messed up. But that's what it would take to have a genetically stable state. And that's what it would take for allele frequencies to remain constant. So this is completely theoretical. And so when we get deviation from Hardy-Weinberg law, we see change in populations. And the cool thing about it is their equation is super simple. Even somebody like me who honestly kind of stinks at math, I can do this. This equation's not that bad. So hopefully you feel encouraged by that if you're in the camp that I'm in, of the we're not so good at math camp. So here's the 5 I gave you. Mutations don't occur, population's huge, it's isolated, mating's completely random, the individuals survive and produce offspring, same exact number. And these conditions are never satisfied, and so allele frequencies are always shifting. And sometimes they shift one direction, and sometimes they come back to normal. But here it is. It's the concept under ideal conditions, and we're going to look at it with flower pigmentation. And so here we have big B coding for blue. Big B, little b is a heterozygote for, I guess, light blue, and little b is white. And so it's a 2 allele system. The gene is flower color. The alleles are big B and little b. Pretty straightforward there. the plants that are homozygous can only make one kind of gamete, which would be, of course, they'd carry big B and big B, or they'd carry little b, little b. The heterozygous can carry two kinds of gametes. It could be a big B or it could be a little b. And so we go ahead and we cross our heterozygous big B, little b, little b, and we see what it looks like in our population. And so if lowercase p, which is the Hardy-Weinberg formula, is p squared plus 2pq plus q squared equals 1, if little p is the proportion of the big B alleles in the gene pool, and q is the proportion of the little b alleles, then the Punnett square shows that each generation in the randomly mating population that they made up and the predicted proportion of the offspring that is going to inherit two big B alleles, that's this guy right here, is p squared. The proportion that will inherit both alleles is 2pq, because there's a pq here and a pq here, and the one that inherits the two little b alleles is the q squared. And so that's where we get the Hardy-Weinberg formula, p squared plus 2pq plus q squared equals 1. And of course, 1 is 100% of the population. And so we will definitely do some work with this. Also, p plus q equals 1 because p is the frequency of this fellow, q is the frequency of this fellow, and so these two guys are the only ones around, so they have to equal 100% if it's just a two allele system. If it's multiple alleles, it would get weird if it were 3 or 4. So genetic equilibrium is used as the benchmark, which is this is no change Hardy-Weinberg law. The further we move from this benchmark, the more we're changing or evolving, even if it's just microevolution. And so they looked at this disease called hemochromatosis, which is HH, and we determined the carrier frequency that we saw this in a lot of people from Irish ancestry. And so what we did was we scanned their DNA and we looked for whether or not they carried the allele. This disease happened to be autosomal recessive. And so we're like, okay, let's just see what the allele, how many people have this allele. And so we got a whole bunch of people from Irish ancestry. We checked out their DNA. We found that it's 14%. Okay, so that's the recessive allele. Doesn't mean they have the disease, but it means they're a carrier. At least they're a carrier for the disease. And so 14% of the people have the allele. So our Q is 0.14. Now, if you don't have this, then you must have the dominant. And so P is just 1 minus 0.14. The rest of the population must be 0.86. Those add up to 1. That's 100%. Cool? So 14% have this allele, which is the recessive one. P is the dominant alleles at 0.86. And so we can figure out how many people in this area are carriers for the disease. And that would be 2PQ. And so we take 2, and you literally just multiply by 0.14 by 0.86, and you get your 0.24 for your 2PQ, because you can do P squared plus Q squared plus 2PQ equals 1, and figure out how many are your heterozygous carriers. If I wrote the AP test, I would have at least one question about Hardy-Weinberg on there. It's a great example of measuring one of the 4 main ideas that the course is built on. So we're going to practice this in class. I'm going to get you guys some work on Hardy-Weinberg. We'll work through them together. And if I can do them, you can do them. Believe me, I am nothing special in the world of math, but it's just pretty easy. So this is Patterns in Natural Selection. We're actually going to stop there, and we will pick that up next time, and which is a really cool talk too. So we'll stop at Hardy-Weinberg. So thank you for your time, and we'll do some